everyone, if you're a first-year MBBS student, I know biochemistry can sometimes feel like a mountain to climb. But today, we're going to tackle one of the absolute cornerstones, how our bodies actually use the food we eat. Let's make the whole process of digestion and absorption crystal clear.
So here's our roadmap. We'll kick things off with the big picture, the body's digestive blueprint. Then we'll follow the journey for each of the big three: carbohydrates, proteins, and lipids. And finally, and this is super important for your exams, we'll tie it all back to clinical practice and do a quick fire recap. Ready?
Okay, before we start chasing individual molecules, let's get one fundamental rule straight. If you understand this one single principle, everything else just kind of falls into place. I just love how our textbook puts this. This little verse, it's not just poetic, it's literally the entire story in a nutshell. We take in complex food, we chop it up into simple little pieces, we absorb them, and then our cells use them for fuel. That's it. That's the core idea we're going to keep coming back to. So what's the official definition of digestion? Well, it's all about hydrolysis. That word is everything here. It means we're using water, hydro, to break, lysis, chemical bonds. We're turning big, clunky molecules into small, absorbable ones. This one chemical reaction is the engine driving our entire digestive system. All right, first up on the menu, carbohydrates. I want you to think about the last meal you ate, maybe some rice or a piece of bread. We're about to follow that food from a complex starch all the way down to the simple sugar that is literally fueling your brain as you listen to this. So the journey kicks off in the mouth with salivary alpha amylase, but honestly, its job is short-lived. The second it hits the stomach's acid, it's inactivated. The real party starts in the small intestine. That's where pancreatic alpha amylase comes in and does the heavy lifting. Then, for the finishing touch, right at the intestinal wall, we have enzymes like lactase and sucrase that do the final snip, giving us those perfect absorbable monosaccharides. Okay, we've got our simple sugars. Now, how do we get them into our cells? Glucose and galactose are clever. They use what's called a co-transporter, SGLT1, to hitch a ride with sodium ions. It's an active process, so it needs energy. Now, this is clinically vital. You know an oral rehydration therapy for diarrhea? The glucose in that solution is what helps the body pull in sodium and, with it, water. It directly fights dehydration. It's a lifesaver. Fructose, on the other hand, is a bit more chill. It just uses a different transporter, GLUT5, and kind of diffuses in. No energy needed. All right, pause for a moment and think about this. We just said that lactase is essential for that final step of breaking down lactose, the sugar and milk. So what do you think happens clinically if someone doesn't have enough of that enzyme?
Exactly. You get lactose intolerance. Without lactase, that lactose just sits in the intestine undigested, and it acts like a sponge, pulling water in through osmosis, which causes diarrhea. At the same time, the bacteria in your gut see it as an all-you-can-eat buffet. They ferment it, producing a ton of gas, and that leads to all the bloating, cramps, and discomfort. Management is pretty simple. You limit milk. But here's a cool clinical pearl. Curd is often okay because the bacteria in the curd have already done some of the work for you.
Okay, next up, proteins. These are the building blocks for, well, pretty much everything in our body. But just like carbs, a whole protein is way too big to be absorbed. It has to be broken down into its individual amino acids first. Protein digestion really gets going in the stomach's acidic environment. The hydrochloric acid makes the proteins unravel, and then an enzyme called pepsin comes in and makes the first cuts. Then the action moves to the small intestine, where a whole team of pancreatic powerhouses, think trypsin and chymotrypsin, take over. The final blows are delivered right on the intestinal wall by other enzymes, which finally frees up those individual amino acids so they can be absorbed. But hang on a second. These enzymes are powerful enough to digest the steak you had for dinner. What stops them from just digesting the pancreas itself? It's a great question, right? Well, the body has this brilliant safety system. It makes them in an inactive form, called zymogens. So how does it flip the on switch only when they're safely in the intestine? And this is just a beautiful biochemical cascade. It all starts with one single enzyme made by the intestinal cells called enteropeptidase. It acts like a key, activating a little bit of trypsinogen into trypsin. Then that new trypsin acts as an amplifier, activating even more trypsinogen. And finally, this big army of trypsin becomes the master activator, switching on all the other pancreatic zymogens. It's a perfectly controlled domino effect that makes sure digestion only happens where it's supposed to. It's genius. And now for our final player, lipids, fats, and their digestion is a unique problem. See, our digestive tract is a watery place, and we all know what happens when you mix oil and water. They don't. So how does the body solve this emulsification challenge? The answer is emulsification. And you can honestly just think of it like using dish soap to break up grease on a dirty pan. The body uses bile salts, which are made in the liver, as its biological detergent. They smash big fat globules into tiny little microscopic droplets. This massively increases the surface area, giving the fat-digesting enzymes, the lipases, way more room to work. So once the fats are broken down, we hit another wall. How do we get these insoluble pieces across the watery layer to the intestinal cells? Once again, bile salts are the heroes. They form these tiny little transport vehicles called mixed micelles. I want you to picture them as little taxicabs. They package the lipids inside and shuttle them right up to the surface of the intestinal cells, ready for absorption. Okay, so the micelle taxi drops its passengers off at the intestinal cell. But once inside, something really fascinating happens. The cell immediately reassembles the pieces back into full triacylglycerols. Now, why would it do that? Well, it keeps the concentration of the pieces low inside the cell so more can keep diffusing in. These brand new fats are then packaged into special transport particles called chylomicrons. And here's the key difference from carbs and proteins. These guys are too big for the blood vessels, so they get secreted into the lymphatic system first. Whew, we've covered a lot of ground. Now for the most important part. Let's connect all this biochemistry to what you'll see in the clinic and recap the biggest takeaways. This is where theory becomes practice. This slide right here is pure gold. You'll want to remember this. Just notice the pattern. For each macromolecule, a specific defect leads to a specific disease. A problem with a carb enzyme, like lactase, you get lactose intolerance. A defect in an amino acid transporter, Hartnup's disease. And if you can't digest or absorb lipids properly, maybe because of a lack of bile, you get steatorrhea, which is just excess fat in the stool. Seeing these connections is absolutely key for your clinical years. All right, time for a quick pop quiz. First question, what is that one key enzyme made by the intestinal cells that kicks off the whole protein digestion domino effect? I'll give you a second. Got it? It's enteropeptidase. Remember, it's the one that turns the very first key. And for our second memory check, what do we call those special lipoprotein packages that are made in the intestine to transport fats into the lymphatic highway? The answer is chylomicrons, the taxis for the lymph system. Awesome job. And just like that, you've done it. You've traced the entire journey of our food, from complex molecules to simple fuel, and you've connected the biochemistry directly to the clinic. You have absolutely mastered a key concept in biochemistry today. Fantastic work. Keep that momentum going.